Alright, so I am going to record today for chapter 5, structure of the major compounds of the body. So just to kind of give a background or a synopsis of why this chapter is important, I want you to think about drug development and currently medications that are on the market that are effective. What makes a drug that is on the market effective, right? Is it the ability of the drug to be membrane selective, so its absorption rate, metabolism? Is it its ability to bind to receptors, whether it is a drug that inhibits a pathway or a drug that activates a pathway? Is it due to its solubility properties, or even if it's a carrier or a transporter type of drug? All of those factors that I just mentioned are actually taken into consideration when drugs are being developed, but it always ties back to the drug's structure. So what is the structure of the molecule or the compound that is being used in this drug development? That is what's important, and that's kind of what we're going to go into in this chapter. It's going to help explain structural components and the chemistry of structures, because one thing I want to point out is structure determines function. So what does the structure of a drug tell you? There's a couple examples here of two different types of drugs, but when you look at the drug itself and you start to break down the functional groups that are present within the drug, it helps you understand how it's going to be absorbed. Is it hydrophilic versus hydrophobic? And also, how is its ability to cross the blood-brain barrier? How effective is it? And so all of those things are important, and you understand these concepts by understanding the structure of the drug. And what makes up the structure of the drug are denoted by the functional groups that are present, whether it's an ether, a carboxylic acid, aromatic hydrocarbon, a tertiary amine hydrocarbons. All of these factors play a role in understanding its function. Right? So the more hydrophobic a compound is, the greater the effect is on crossing the blood-brain barrier, and that usually indicates lipid solubility. So we have two cases here, diabetes and lidotopane. I'm not going to go into detail about these. You can read them, and I'll give you a second. You can pause the video at this point to read on them. The new patient that we have is lidotopane. So that's the one that I'm going to just read or give an overview of. She's 47 years old. She came into the office complaining about a severe throbbing in the right great toe. That began about 8 hours earlier. The toe had suffered from no trauma, but it appeared red and swollen. So she had no physical trauma that occurred to the toe, but it was showing signs of inflammation, right, due to the redness and the swelling. It was warmer around the surrounding tissue and very tender with light pressure. Ms. Topane is unable to flex voluntarily or extend the joints of the digit and passive motion, which causes great pain. So thinking about those Miss Diabetes, who we've been following her since we first started in chapter 1, and now we're introducing a new patient, Miss Topane. This is hopefully helping you visually. Miss Topane will help you visually see the effects of what we're going to talk about in this chapter, and how potential factors of functional groups contribute to various diseases. Okay, so what role does functional groups have on biological compounds, right? Well, one, you have to understand the basic composition, okay, which we're going to talk about. We're also going to go and talk about in this chapter some functional groups and the polarity of bonds and the partial charges. When I say polarity of bonds and partial charges, I'm referring to its effects on solubility and reactivity. And then we're also going to talk about nomenclature. Okay, so now getting into biological compounds. When we talk about biological compounds, I want you to think about any molecule or compound that is composed of carbon, hydrogen, oxygen, nitrogen. Those are all examples of elements or molecules that make organic compounds in the body. You see phosphorus in DNA. You see sulfur in certain proteins. You see nitrogen in nucleic acid. So these are various components that are made up of various compounds in the body. The key component is carbon. Okay, and carbon is one of those molecules that is able to form covalent bonds with other molecules. So carbon is really a central molecule for most biological compounds. Okay, carbon can be a carbon atom can be joined by a single or double bond to form what we call a carbon backbone. Okay, and the number of carbons and hydrogens are referenced by the number present. So ethyl refers to 2, methyl refers to 1 carbon, butyl refers to 4, and etc. Okay, we also see here on the right side of the screen where you see this 6-ring structure. It's called an aromatic phenyl group or a benzene ring, and it has 6 carbons, 2, 3, 4, 5, 6. And in this aromatic phenyl group or benzene ring, there are 3 double bonds. Okay, but then you have an aliphatic isopentyl group, which has a single carbon carbon bond, and a double carbon bond. The presence of these various molecules and their ability to engage in various bonds also is incorporated into their name. So a lot of molecules names are derived from the actual composition of molecules within it. For example, if you see ene or ene, which you see here, it denotes the presence of a double bond in the molecule. Okay, ene denotes the presence of a double bond in the molecule. If you see iso, it refers to a branched molecule. Or if you see aliphatic, that means there's no ring, right? And so the nomenclature of a molecule denotes its structure. If you see like ene in it, you should know that that's most likely like a benzene ring. Okay, it has 6 carbon ring structure in there, and sometimes it'll be referred to as a phenyl or phenol group. So what is it about functional groups, right? They really are essential in many biological processes that happen in our body, but they are made up of, most of them have a carbon skeleton, and attached to their carbon skeleton or backbone, they have functional groups that are attached. And these functional groups have the ability to react with other molecules. Okay, and so this helps to understand how you can have reactivity in the body. Remember, on last chapter we talked about reactivity, polar versus nonpolar, dipolar, and how water interacts with CO2 in the body through metabolic processes to form carbonic acid. Okay, so these are all very important factors. But the presence of carbon-carbon and carbon-hydrogen bonds and their ability to share electrons equally help determine whether the bonds are nonpolar or relatively unreactive. So in bonds with carbon and oxygen and carbon and nitrogen, electrons are going to be shared unequally. This means that, as we talked about last week, the bonds are going to be polar and more reactive. Okay, so some will be more electropositive versus electronegative. And as a result of having this unequal sharing of electrons, which causes reactivity, these functional groups can be more reactive due to their proportions of availability of reactivity, right? So it'll determine the type of reactions that take place. It also affects their physiological role in the body. One molecule that we are familiar with is ketone bodies, right? We've talked about ketone bodies. Well, when you look at ketone, one word in there is O-N-E, one, right? And we see that in acetone. Right, so acetone is a ketone. Ethanol has OL at the end. Well, OL at the end of a molecule often denotes an alcohol, right? An acyl group, it has a proportion or a portion that has a carbonyl group, so a C double bond O, right? And an ester or an amine linkage, and you'll often see at the end of that name a YL. Okay, so ultimately what I want to point out here is when you have names of molecules, you should be able to look at the name of a molecule and identify what functional groups are present. Okay, what functional groups are present by looking at the name, because it's highly indicative of the functional groups that are present and potentially its reactivity. Okay, for example, 3 fatty acids that are esterified to glycerol, that lets you know that there's a common compound that has 3 alcohol groups. Okay, OL, alcohol. So these next 2 slides, I believe, you're able to see the different functional groups. Now you all probably would have had this hopefully if you've taken organic chemistry 1 or 2, this should not be familiar to you, because these are components that you've talked about before, that you've seen before, whether you're talking about an aldehyde group, an alcohol, ketone, a carboxylic acid, an amine group, a phosphoester bond, an amide. These are all very important functional groups that are present on various compounds within the body. Okay? Now at home, I want you all to pause this video and I want you to do this question. I want you to answer this question, think about it, read it, and then be able to reflect on this and write it down, because you'll see something like this again on one of your exams. And this is the answer. Now let me go back. What it was actually, the question was asking you is, what functional groups are present in each of these ketone bodies? These are 3 ketone bodies that we've already seen before: beta hydroxybutyrate, acetoacetate, and acetone. You should be able to look at these 3 different molecules and identify what functional groups are present. Right? So beta hydroxybutyrate, even if you couldn't look at the actual visual name structure, but the name gives you an indication. The ATE, dissociated carboxylic acids, acetate, and acetone, let you know there's ketone groups in there. Okay? Hydroxybutyrate, hydroxyl, there's a hydroxyl group there, alcohol. So looking at the name of the structure should tell you what functional groups are present. But if you can't look at solely the name, go back to the actual visual representation and look at the various components so that you can identify the various carboxylic acid, the various functional groups that are present. Okay, another question, pause so that you can answer this question. And it is, which compound is glycerol and which compound is glyceraldehyde? So you don't know which compound, A or B, is glycerol versus glyceraldehyde. However, you should be able to look at the structure and identify what's present. Well, we know that if anything ends in OL, it has an alcohol. Well, how do we know it's an alcohol? It has an OH, hydroxyl group. Okay? If you see aldehyde in it, that means it has an aldehyde group. An aldehyde group has a carbon-oxygen double bond attached to a hydrogen. So in this case here, we don't see an aldehyde group, but we see an aldehyde group here. So this has to be glyceraldehyde. We see 3 hydroxyl groups, so that has to be glycerol. These are things that you want to look at and break down so that you understand the actual structure determines function. Okay? So just kind of breaking this down, we talk about oxidized and reduced. Okay? Whenever you have talking about oxidation, it's referring to the number of electrons around a carbon molecule. Okay? So in oxidation, you have the loss of electrons and the loss of hydrogen atoms with 1 or 2 electrons gained or lost. So it's gain or loss of oxygen, and usually in the earlier stages. Reduction is the gain of electrons and results in the gain of hydrogen atoms or loss of an oxygen molecule. Okay, so an easy way to say this is, when you're looking at oxidation, it is going to be the gain of an oxygen or a hydroxyl group. Reduction is the loss of an oxygen atom. Okay, carbon-carbon double bonds are more oxidized and less reduced than carbon-carbon single bonds. Let me say that again. Carbon-carbon double bonds are more oxidized and less reduced than carbon-carbon single bonds. Pause again. Here's another question that I want you to be able to answer on your own. And here is the answer. Okay. The breakdown of this is just showing you that acetoacetate is more oxidized than beta hydroxybutyrate. And the way to determine this is by looking at the number of the presence of carbon-carbon double bonds versus carbon single bonds. Beta hydroxybutyrate is more oxidized. Acetoacetate is more oxidized than beta hydroxybutyrate. Now, remember, when we looked at the term of oxidation, okay, it's the gain of an oxygen atom or hydroxyl group. But carbon-carbon double bonds are more oxidized. So when you look at 2 molecules, look at which one has more carbon-carbon double bonds. If it has more carbon-carbon double bonds, it's going to be more oxidized. So because acetoacetate has more double bonds, it's going to be more oxidized than beta hydroxybutyrate. All right, so thinking about groups that carry a charge. Acidic groups contain a proton that can dissociate, and it usually will leave the remainder of the molecule, an anion, with a negative charge. Okay? In biomolecules, the major anionic substituents are carboxylate group, phosphate groups, and or sulfate groups. Phosphate groups are attached to the metabolites and are often abbreviated with a P, with a circle around it, or just the P, as we see with glucose 6-phosphate. So some examples of major ionic substituents are carboxylate group, a phosphate group, a sulfate group. That 8 in the word denotes a negative charge. So whenever you see a carboxylate, phosphate, sulfate, know that it's going to carry a negative charge with it. Okay. Also, compounds that contain nitrogen are going to be basic, and they're going to acquire a positive charge. So these are just some characteristics to help you remember or to think about these molecules when you see them, because it's going to again tell you about its ability to interact or to respond. So let's look at nitrogen for an example of understanding charge. Nitrogen has 5 electrons in its valence shell. You all should remember the valence shells and electrons from Gen Chem. Okay, if only 3 of these electrons form a covalent bond with other atoms, then nitrogen has no charge. Okay, if the remaining 2 form a bond with hydrogen or carbon, the nitrogen carries a positive charge. So looking at, let's say, amines consist of nitrogen attached through a single bond to hydrogen atoms or to 1 or more carbon atoms. A primary amine has 1 carbon-nitrogen bond. An example will be dopamine. It's a weak acid. We learned about that in last chapter, in chapter 4, and it has a pKa of approximately 9. So we know that it is a very weak acid. Okay. At a pH of 7.4, it carries a positive charge. So this brings us to understanding the polarity of bonds and charges. Okay, the polarity of bonds and its charge molecule helps us fully understand solubility and reactivity. The solubility helps you understand how a molecule is going to react with water and other molecules. The reactivity is going to help you understand how the molecule can interact biochemically with other compounds. So the charge that it possesses and how it's going to interact. So solubility determines how it reacts with water and other molecules. Reactivity determines or helps you understand how that charge will engage in a biochemical reaction. In polar bonds, i.e. covalent bonds, the electron cloud is more dense around one atom versus another. So this is going to make or result in a molecule being more electronegative than electropositive. So when we look at oxygen is more electronegative than carbon. So when you have a carbon-oxygen bond, it's polar. Okay, oxygen is carrying more of a partial negative charge. The carbon is carrying a more partial positive charge. This is how you get a dipolar molecule. Now, in a nonpolar carbon-carbon or carbon-hydrogen bond, there are 2 electrons in the covalent bond, and they're shared equally. Okay. Now, if you incorporate nitrogen, it's going to carry a partial negative charge. So, because of that, carbon-nitrogen is polarized. Sulfur, on the other hand, can carry a partially negative charge. And so, when you're looking at these different molecules here, it helps you to understand reactivity, but also solubility. Okay, so solubility is the ability of a molecule to interact with water, primarily with water, right, or in an aqueous environment. In order for a molecule to be soluble in water, it must contain a charged or polar groups that can associate partially. So having partial negative, partial positive charges, like we saw with last chapter with CO2 and water. So how is solubility determined? The proportions of the polar to non-polar groups in the carbon backbone, okay, in that carbon skeleton. When you look at polar groups or molecules, they tend to be hydrophilic, okay, so they are able to interact with water. Non-polar groups within a molecule are hydrophobic, so there's little to no interaction with water. You have glucose 6-phosphate, which you're seeing here. You all should know glucose 6-phosphate from the process of glycolysis. Well, glucose 6-phosphate contains polar groups, very hydrophilic and water soluble. Okay, very polar and water soluble molecule. So because of that, if you were to place it in the aqueous solution, they're going to cluster together. And when they cluster together, they can form what we call either van der Waals interactions, which you kind of see with the gecko climbing up a wall, right? That interaction is the van der Waals forces, or hydrophobic interaction. What is the hydrophobic effect? This occurs when compounds are like pushed together and water employs this energetic reaction, and it forms like a lattice, okay? Lipids that are placed in the aqueous solution start to come together, aggregate, and they form these droplets because they're repelling that aqueous solution or that water. Okay, so example would be like vegetable oils and salad dressing. Now with reactivity, reactivity is a consequence of polarity. And so when you're looking at molecules that have partial charges, right, this allows for them to be reactive. Right? These charges, whether they're partial or full, dictate that there is the potential for a biochemical reaction to occur. This would be back to those concepts you learned in Gen Chem about electrophilic and nucleophilic attacks. That partial charge on carboxyl carbons attract partial negative groups, and they form reactions including carboxylic acids. So how do we identify this carbon chain or this reactivity? The first carbon that you see in the compound, okay, that is the most oxidized, is number 1. So in this case, remember we talked about what is most oxidized. It's the presence of a double bond. So this has the most oxidized state. So this would be your first carbon. The second carbon from that is going to be considered carbon number 2, but it's also the first carbon that's given a Greek letter, which would be alpha. And so then the third or the next one over from that carbon would be beta and so forth. So you can either count numerically 1, 2, 3, 4, with number 1 being the most oxidized carbon, and then thereon after, or after that first oxidized carbon, the next one is considered the alpha. So if we were to look at that nomenclature or those rules, look at 3-hydroxybutyrate, okay? 3 off of the third carbon. Okay, you have a hydroxyl group, which you see here. Butyrate, but means 4, so 1, 2, 3, 4. There's 4 carbons. Okay, and so that should hopefully help you understand the nomenclature of naming molecules. Okay, the first carbon is going to be considered identified based off of its oxidized state, and then using the Greek letter will be the first carbon after the most oxidized will be alpha. Okay. Here are all of, again, those different functional groups here to help you. And you can see the formation in this diagram here, the formation of esters, thioesters, amides, phosphoesters, and anhydrides, which you see here. All right, carbohydrates. And at any time you're listening to this, I'm trying to not be too long, but you can always pause. If there's something that I cover that you have more questions on, please feel free to send me an email or come by for office hours to get more clarity. This chapter is really going to focus a lot on structure. So understanding the structure to help you understand function. So you'll need to know how to identify molecules. There's no other easier way to do it than just to practice by reading, looking at different molecules, identifying the functional groups, and being able to answer those questions. So it is a lot of memorization of functional groups. All right, so carbohydrates. You have what we call simple monosaccharides, and they're going to consist of a linear chain of 3 or more carbon atoms, one which forms a carbonyl group through a double bond with oxygen. Okay, the other carbons of an unmodified monohydrate contain hydroxyl groups, and they result in the general formula for an unmodified sugar as CH with a 2 subscript O. Okay, and you usually see OSE in the name, in the suffix of the name. Examples: ketose. Okay. If you have a carbonyl group, it's an aldehyde. The sugar is an aldose. If the carbonyl group is a ketone, you have the sugar that is a ketose. Okay. So here you have a D and an L sugar. The D and the L sugars denote the location of the hydroxyl group on the left or the right side of the carbon backbone. This denotes the stereoisomers and the epimers. Hopefully you all remember about them. We're going to go into that a little bit more, and we'll also talk about the ring structures, the substituted sugars, and so forth. And we'll also talk about glycosides. So let's go into monosaccharides. As I kind of said earlier, very simple chain, 3 or more carbons. The one carbon forms a carbonyl group with a double bond with oxygen. The other carbons are going to contain a hydroxyl group, as I previously said. And attached to it will have in the suffix OSE. Now, I gave the examples already of ketose and aldose. However, you can look at the name and it's going to help you understand how it's classified according to the number of carbons. So 3, 4, 5, 6 carbons. Okay, triose, tetrose, pentose. Fructose is also called ketohexose. Glucose is also called aldohexose. So think about sometimes when you're reading certain ingredients of molecules from a medical perspective, you may not see glucose, but you may see aldohexose or dextrose. Okay, and that also has to do with the location of it. So when we talk about D and L sugars, they are monosaccharides, and they can have the same composition, but symmetrically they're different. Those are stereoisomers. So they have the same chemical composition, same number of carbons, nitrogen, hydrogens, but how they differ is the symmetry. So the location of some of these functional groups on the carbon skeleton or the backbone. So what are stereoisomers? Stereoisomers are compounds that have the same chemical formula, but they differ in position of the hydroxyl groups. And so it causes them to be asymmetric in their carbon backbone. Okay, so they have an asymmetric central or chiral. These groups attach to the asymmetrical carbon atom, and they can be arranged in 2 different forms, almost like mirror images of each other. Okay, so when you have monosaccharide stereoisomers, they are designated as either D or L, and that again is based off the position of the hydroxyl group farthest from the carbonyl carbon. Okay, so here we have D-glyceraldehyde, L-glyceraldehyde. Notice on the D-glyceraldehyde, it's on the right side, L-glyceraldehyde is on the left, and these are mirror images of each other. If you were to take a mirror and put it in between the two, they are mirror images of each other. The L form has the hydroxyl group on the left, the D form has the hydroxyl group on the right. Now, what's the relevance of this D versus L, right? We still use it in medicine today to describe sugars and even amino acids. The D series is what most sugars in humans tissue belong to. So you'll see D-glucose, it's dextrose, okay? And that's what you see denoted in medicine oftentimes. It's dextrose. If you see dextrose on any ingredients, it's referring to the D form of glucose. Okay, so stereoisomers and epimers. So we've already talked about stereoisomers, but epimers, what are they? Epimers are stereoisomers, but they differ in the position of the hydroxyl group at only one of their asymmetric carbons. So you have D-glucose, D-galactose. Those are epimers, but they only differ at position 4. So D-glucose, D-galactose. They differ at position 4. 1, 2, 3, 4. 1, 2, 3, 4 is where they differ. Okay, so on one case the hydroxyl group is on the right, the other the hydroxyl group is on the left, and that's how they differ. The same exact chemical composition, but the hydroxyl group differs on the position. Okay, they can be interconnected in human cells and by enzymes, and they're called epimerases. Okay, so epimerases are the enzymes that catalyze that inversion. D-mannose and D-glucose are epimers, and they differ at position 2. So 1, 2. hydroxyl on the right, 1, 2, and D-mannose, hydroxyl on the left. Now, another important factor that I want to denote is the reason why they can be chemically the same but structurally different, and in part is how they react when you put them in a solution. So their reactivity, right, will cause them to have a structurally be different. Okay, so whether it forms a closed ring, 6-ring structure, or if it doesn't, depending on the environment it's placed in, and that will cause it to react differently. So in solution, monosaccharides exist as ring structures, as I previously said. The carboxyl group reacts with hydroxyl group at the same molecule to form that 5-6 membered ring structure. The hydroxyl group at the alpha position, okay, this is the first one, so that means according to the nomenclature, the next carbon, the second carbon, should be the alpha carbon, and you'll see here between D-glucose and D-fructose reacts differently. Okay, it moves to the B position. The oxygen becomes part of the ring structure, and the ring structure can be changed from the alpha to the beta position. So it's the ring structure changing, okay, from alpha to the beta position alters its structure. Same chemical composition or similar composition chemically, 6 carbon structure, but the positioning of the functional groups will alter how it responds and how the structure it creates. All right, substituted sugars. Again, going back to kind of what we've been talking about with epimers, sugars can frequently contain phosphate groups, amine groups, sulfate groups, or even acetyl groups. I'm sorry. Most of the free monosaccharides within the cells are phosphorylated at their terminal sugars or carbons. And that will prevent transport out of the cell. So when you look at different molecules, you can have them to be marked or modified differently, and it helps to regulate function. Okay, so a lot of these sugars can be further modified, and further modifying these sugars will alter how they move, where they're located. So for some, if they're phosphorylated on their terminal sugar, they will not be able to move out of the cell, so it causes them to be arrested within the cell. An example would be glucose 6-phosphate. So being able to modify these molecules can also determine or regulate their function. All right, so sugars can also be oxidized at their aldehyde carbon to form an acid. So once that happens, technically they're no longer a sugar. The change in the name should also be denoted. So they'll go from OSE, denoting a sugar, to onic acid or onate. Okay, you see that with let's say a carbon that contains a terminal hydroxyl group, and it becomes oxidized. The sugar is now called uronic acid. If an aldehyde is reduced all the way, or all carbons contain hydroxyl groups, then it'll also have a change in the nomenclature. If you have one sugar that is reduced, leaving the carbon with a hydrogen, that sugar is now deoxyribose. Hopefully you all should think about what comes to mind is DNA. Deoxyribose, right? Nucleic acid, DNA. So remember, on the ribose, the sugar in DNA, attached to that phosphate group, you have that second carbon. That second carbon does not have an oxygen attached. It just has a hydrogen. And so that's where deoxy comes from, without oxygen. Glycosides, okay? The hydroxyl group on the anomeric carbon of a monosaccharide, it can react with hydroxyl group or an amine group of another compound, and together they form a glycosidic bond. So the amine group of one molecule and the hydroxyl group of another can form a glycosidic bond. Hopefully you all, something comes to mind when you hear that from genetics. Okay, the N-glycosidic bonds are found in nucleotides and nucleosides. Ultimately, when you see the this happening, you have, or I would say in contrast, okay? So in contrast, you have O-glycosidic bonds, such as like lactose, and they join sugars to each other or attach sugars to hydroxyl groups of an amine acid on a protein. So this should hopefully stimulate or bring back to your remembrance when you learn about proteins and the formation of amino acids in your undergrad or even some of your genetic classes. So that's what this is visually showing you, that reaction of the N and O glycosidic bonds. All right, so disaccharides, oligosaccharides, and polysaccharides. So break down the term that you see here, disaccharides, di being two. So disaccharides are two monosaccharides that are joined together by an O-glycosidic bond. The example, as I said earlier, is lactose. Oligosaccharides are 3 to 12, so it's a typo, 3 to 12 monosaccharides linked together. They are often found attached to a nitrogen or a O or N or O glycosidic bond to form proteins called glycoproteins. Okay, when you hear polysaccharides, it could be 10 to thousands of monosaccharides that are joined together to form a linear or a branched structure. So here, when you have lipids, there are different types of lipids. Lipids is a general term, but you could have fatty acids, acylglycerols, phosphoacylglycerols, sphingolipids, steroids, and they all have different roles. And how they are named is based off of, again, their structure. Fatty acids are usually aliphatic, linear, with a methyl group at one end and a carbonyl group on the other. Carbonyl group, methyl group. In humans, fatty acids have an even number of carbon atoms, and they're usually between 16 to 20 carbon molecules. And we can further denote them or classify them by saturated versus unsaturated. Saturated fatty acids have a single carbon bond in the chain. The most common is palmitic acid, stearic acid. Palmitic acid has 16 carbons, stearic acid has 18 carbons. Unsaturated fatty acids have more than one double bond. And with unsaturated fatty acids, I want you to think about how they differ from saturated fatty acids. Now, some shorter fatty acid names are based off of the number of carbons. So octanoic acid, meaning it has 8 carbons, oct in front of it. Decanoic, dec is 10, so it has 10 carbons in it. But ultimately, when you think about these fatty acids, look up their composition and ask yourself, how does their composition associate with its function? Well, when you think about fatty acids, I want you to think about melting point, because that has a major factor, and this also ties back to the presence of carbon bonds. Okay, the melting point of a fatty acid will vary, and it's based off the number of double bonds present. So the melting temperature will increase with the chain length, and it'll also decrease with the degree of unsaturation. What do we mean by that? Okay, the fatty acids with many double bonds, such as like vegetable oils, they're liquid at room temperature. And saturated fatty acids, such as those like butter, fat, they're solids at room temperature. So lipids, with lower melting points are more fluid at body temperature and contribute to the fluidity of your cellular membranes. So monounsaturated fatty acids, one double bond. Polyunsaturated fatty acids, 2 or more double bonds. That position of the double bond is designated by the number of carbons in the double bond closest to the carbonyl group. So oleic acid has 18 carbons. The double bond is positioned between 9 and 10, and so we denote its nomenclature, or its name is 18:1 delta 9, because that is where the double bond is present. I wish I don't know if I have another example here for you. Here we go. Okay, so this would be where the double bond is located. So let's go back for a second, because I think I have some examples here for you. Actually, let's just go on to these so I can help break down how you name them. Now, you're going to most often see the molecules drawn like this, but but I want you to know that these points are denoted as the presence of a carbon, as you see here. Okay, so here's your carbonyl, here's your methyl group. So you're always going to start the nomenclature, the naming and the numbering at the carbon here, because it is the most oxidized. Remember that, we learned that, slides back. Okay, so this is going to be your first carbon. Now remember, after that first carbon, that second carbon is where your alpha or your Greek letter starts. So that's where alpha will be, and then beta, gamma, delta, and so forth. Okay, so this is how the first part of how you are naming your fatty acids. Count the number of carbons: 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. So this is an 18 carbon molecule. So 18. The next number after the colon denotes how many carbon double bonds are present. Okay, there are no double bonds here. We're not counting this carbonyl. So there are no double bonds here, so it's 18 and 0. Now here we have a double bond. It's still 18 carbons, but there's one double bond, so it'll be 18 colon 1. But however, remember, we have to now denote where the double bond is located. So you're going to put delta superscript, and then you're going to have to count to where you see the double bond. So 1, 2, 3, 4, 5, 6, 7, 8, 9. So your answer should be delta 9, or you may see it referred to as omega 9. Okay? Now, here, alpha linoleic acid, you're going to look at how many double bonds are present. But here we have 3 double bonds. Okay? So because we have 3 double bonds, we now need to be able to number them. So we know that it has alpha linoleic acid has 18 carbon. There are 3 double bonds. And so you're going to have delta 9, and then you're going to put how many, or the carbon number associated with where the double bond starts. So here we know the first one is 9, 10, 11, 12. So delta 9, delta 12, 13, 14, 15, delta 15. Okay? And so then this becomes omega 3. All right? So I want you to pause it here and do this question. Okay. So hopefully you were able to read this question, and it's ultimately asking you about these 3 acids, and it says palmitic acid is a 16,1 delta 9 fatty acid. How would you name it as an omega fatty acid? Now, let's go back to this diagram here. The omega is going to start from the end, right? Omega. And then you're going to count how far away from the end do you have a double bond. And that is your omega. So in this case here, for oleic acid, you go back to the end of this carbon, the omega carbon, 1, 2, 3, 4, 5, 6, 7, 8, 9. It is 9 carbons away from where the first double bond appears, so it is omega 9. In alpha linoleic acid, here's the omega, the last carbon. We're going to count till we get to the first one. 1, 2, 3. So it is omega 3. It is 3 carbons away from where the first double bond appears. Then you have arachidonic acid, 20, right? 1, 2, 3, 4, 5, 6. So it's omega 6 because it is 6 carbons away from where the first double bond appears. So back to this question with palmitoleic acid, if you count it, it is an omega 7 fatty acid because it is 7 places from where the first carbon bond appears from the last carbon, or the omega carbon. I hope that helps you with the classification of the omega fatty acids and the nomenclature of how you classify the carbons along with delta. All right, so acylglycerols. Okay, an acylglycerol comprises glycerol with 1 or more fatty acid. The key here is acyl, or an acyl group. which is attached through ester linkage, as you see. The number of fatty acids esterified to glycerol determines its name. So monoacylglycerol, one fatty acid is attached. Diacylglycerol, two fatty acids are attached. Mono being one, di being two. Phosphoglycerol, it contains a fatty acid that is esterified to positions 1 and 2 of glycerol and a phosphate. If only a phosphate is attached to the carbon 3, it's phosphatidic acid. Phosphatidic acid serves as a precursor for synthesis of phosphoacylglycerols. Okay, you have phosphatidylcholine. It's one of the major phosphoacylglycerols that are found in membranes, and it's due to its amine, which is positive charge, and its phosphate charge, which is negative. So it is amphipathic. Sphingolipids. Hopefully you all are not new to sphingolipids, but sphingolipids do not have a glycerol backbone. They are formed from sphingosine, and sphingosine is derived from serine, a specific fatty acid, palmitate. Ceramides are amines formed from sphingosine by attaching a fatty acid to an amine group. Ceramides and amines, various sphingolipids are produced from the addition of different groups to the hydroxyl group on the ceramide, which you see here. Sphingomyelin contains a phosphocholene group attached to a ceramide and is a component of cell membranes in the myelin sheath around neurons. So sphingolipids are important. Okay. Steroids. Steroids contain a 4-ring structure. They are called the steroid nucleus. Cholesterol is the precursor in human cells from which all steroid hormones are synthesized. Okay, and it's just by modifying making modifications to that ring structure or that carbon 20 side chain. Now, keep in mind, cholesterol is not very water soluble. Is converted to amphipathic water-soluble bile salts such as cholic acid. But these bile salts line the surface of lipid droplets called micelles. Okay, we've learned about this before in the lumen of the intestines, and they keep the droplets emulsified in an aqueous solution. So they are important. Make sure I'm not forgetting anything that I want to point out here. All right. So nitrogen-containing compounds. This should not be something new for you all. If you have taken your genetics, you've taken cell bio, you've taken physiology, you know that nitrogen-containing compounds are important. The presence of nitrogen in an organic compound will increase its solubility in water, right? Whether nitrogen is charged or uncharged. There are different types of nitrogen-containing compounds, whether it's amino acids, you have your purine, pyrimidines, and pyridines, nucleosides, nucleotides, and even tautomers. So, amino acids. Let's get to amino acids. These are important. They're important molecules because they are the building blocks for proteins. They have different configurations. In proteins, you'll see the L form, like alanine. Non-protein, like in bacteria, D amino acids, and you'll see this used in the cell wall. And oftentimes you'll see others, like in other forms, like seen in neurotransmitters. The L form is the amine group that is attached to the alpha carbon. Okay, right here. Purines, pyrimidines, and pyridines. This is what you see in nitrogenous bases. They're the 3 main common types, like adenine, thymine, and then you have it like B6. Niacin. These are part of the purine pyrimidine, which are nonpolar and hydrophobic, and they tend to be relatively unreactive because all of the electrons are distributed equally. Very straightforward. The nucleosides and nucleotides are a part of the backbone, or are the structure, I should say, of DNA. The nucleoside is a base joined with a sugar. There is no phosphate attached. And remember, we just talked about this. These are joined together. The sugar and the base, okay, are joined by an N-glycosidic bond right here. There's the nitrogen. Okay, a nucleotide has three components: a base, a sugar, and a phosphate. So this would be a nucleotide, all three. Okay, ATP, which has adenosine in it, has the addition of the ribose and is indicated in the name change. Okay, it has a glycosidic bond present. So you see here, glucose. The number of phosphates are denoted in the name, tri. Okay, nucleotide triphosphate. So there are three phosphates attached. Tautomers. These are molecules that have a shifting in the chemical structure of the molecule, and it can, in tautomers, due to the shifting, it can alter how these molecules react and respond. So you have, let's say, a nitrogen and a carbon double bond with a hydroxyl group. You can have a shifting in the structure where the nitrogen is bound to the hydrogen, but the carbon and the oxygen now share a double bond. Okay? They're technically the same molecule, but because they have a change in the chemical bonds that are in its reactivity between the molecules within it, it will act differently. And we see this oftentimes like in DNA as well, and it can cause genetic mutations. One portion of the compound may be more reactive than the other, and so it can cause various chemical shifting that takes place and allows the molecule to act as a different molecule and respond differently. All right, so the last portion that I want to kind of talk about is free radicals. These are compounds that have a single electron in the outer orbital shell that allows it to be reactive, and they can exist independently in a solution, or they can be in a lipid environment. Now, many compounds that we have been exposed to are capable of being converted to free radicals. Usually when we talk about free radicals in the medicine or medical field, it is reactive, and therefore it is important physiologically, because that means if it is reactive, it has the ability to stimulate a reaction that can be detrimental internally. Okay, so some side effects of free radicals are it can induce apoptosis. Free radicals can cause tissue damage, and that can, over time, can lead to chronic diseases like diabetes, arthritis, stroke. So when we talk about free radicals, it can be very deleterious, and it's maybe seen as something really small initially, but it has the ability to create a reactive component, which is what you don't want to happen. All right, so here's another point in the lecture where we're almost done. You can pause to read this question. If you're back after pausing, reading through this question is asking you about our first patient when we open up with lidocaine. And it says, judging from the similarity in structure, do you think it's derived from degradation of purines, pyrimidines, or pyridines? Now, in order to really answer that question about uric acid, okay, you have to look up or think about the structure. So uric acid contains two rings, and it's similar to purines, adenine, and guanine. When you look at it and think about urinary excretion, the product formed from oxidation of these two are purine bases. It's not very water soluble, as specifically a specific pH near the pKa of an acidic hydroxyl group. If in excess amounts, if this is present in excess amounts, the sodium urate tends to precipitate in the joints, causing the severe pain of gout experienced by Ms. Topane. And so this is what you're experiencing. Okay, that's the response that you want to look for. So thinking about how uric acid structure can accumulate. Okay, if it is not excreted properly, it can accumulate, and because it's not water soluble at a specific pH, it can start to precipitate and cause pain in the joints. And this is what we're seeing with Ms. Lidocaine, and it resulted in gout. Okay, so this is another important clinical association with the biochemical experience of uric acid buildup. Also, I want you to read through the reducing sugar test. For those that don't have the textbook, I've provided for you. But I want you to read through this because it's also going to help you with understanding how various tests work. A lot of times, when you take various tests, you don't understand the biochemical reactions that are taking place to produce a outcome that is measured to make a diagnosis. So make sure you go through this one. All right. And last are the two overall or clinical comments about diabetes and lidocaine. I will most likely ask some questions about diabetes. So I want you to make sure you pay attention to this as well as lidocaine. That is chapter 5. The take home from chapter 5 is you will experience questions about a lot of nomenclature. So please take the time to go over nomenclature because you will see that again.